Hello and welcome yet again to another very entertaining video. Today we want to talk about insulin and its metabolic syndrome with its associated manifestations, type 2 diabetes, non-alcoholic fatty liver disease, even heart disease and atherosclerosis are all kind of focused on the key aspects of this one hormone, insulin. This is an important one. This is critical to understand. Now, to be sure, there's still a lot we don't know about insulin. But in this video, I'll do my best to kind of help you be oriented to where you are, what's going on, and how insulin could have such a huge impact on your body's health. So we're going to start today with a little anatomy lesson, and we're really focused on the pancreas down here. But here you can kind of see it in context. We've got the liver, we've got the gallbladder. Of course, the gallbladder dumping its contents, bile, into the small intestine, and it shares the common bile duct with secretions from the pancreas. Now, we've talked in our digestive unit about the pancreas releasing things like bicarb to help buffer pH, enzymes to help break down carbohydrates, lipids, proteins. All of these are an essential component of what the pancreas does. However, if we zoom in and take a look at the pancreas, we'll see little pocks, these clusters of specialized cells within the overall makeup of the pancreas. These are called islets of Langerhans. And this is where we want to focus our time in this video. So let's zoom in on one of these. I'm actually going to find at least 5 different cell types in these islets. We'll start at the top. These yellow cells we'll call alpha cells. The majority of cells are called beta cells. We have 3 other cell types. We don't want to spend too much time on these. Delta cells, gamma cells, and epsilon cells. Now, each of these has a separate and distinct purpose. Like we mentioned a minute ago, the beta cells make up the majority. This is about 70% of the total population, and these beta cells are responsible for the production and release of insulin. The alpha cells are the second most populous at about 20%, and they're responsible for the production and release of glucagon. And then that means the rest of these together make up the remaining 10%. The delta cells make and release somatostatin, which, as we've discussed previously, is kind of a pan inhibitor of a lot of things. The gamma cells make and release a little protein called pancreatic polypeptide, and the epsilon cells are releasing ghrelin. However, these 3 bottom cell types are not the focus of this presentation, and what we want to turn our focus to now is on the alpha cells and the beta cells, and understanding how glucagon and insulin work together to maintain glucose homeostasis. So let's talk for a minute about insulin. Insulin is a protein. We've got a gene for it. We make the mRNA, process that through the rough endoplasmic reticulum, modify it in the Golgi, all of that stuff that secreted proteins go through. The thing I want you to associate with insulin is this. Insulin is an anabolic hormone. In other words, it's necessary for growth. It does this through the effects of carbohydrate metabolism, lipid metabolism, and protein metabolism. So let's look at each of these individually. Starting with carbohydrate metabolism, there's 3 key things that we want to make sure we understand. First is the increase of glucose transport, and this is specifically into skeletal muscle and adipose. And we'll show some more specifics on that in just a minute. The second thing we want to think about is increased glycolysis. And it does this actually by increasing enzyme activity. We know of at least 2 enzymes that are associated with this. This is going to be hexokinase, which makes sense as this is the first reaction in glycolysis, converting glucose to glucose 6-phosphate. And then, of course, the key regulatory enzyme for glycolysis, which is phosphofructokinase. And then the third thing we want to think about with carbohydrate metabolism is increased glycogen synthesis. If insulin is up, it's because I have an abundance of glucose, and therefore it's a great time to store glucose in the form of glycogen. All right, let's look at lipid metabolism. First and foremost, we see increased fat synthesis commensurate with decreased lipolysis. This, of course, has the general effect of lowering my blood or my plasma free fatty acid concentration. What it also means is I'm not burning as much fat. So the second thing we want to think about is decreased beta oxidation. All right, then something that's commonly overlooked. Let's look at protein metabolism. Here we see increased amino acid uptake, which leads to increased protein synthesis. This is primarily in muscle. but we'll also see it in adipose, liver. And of course, if we're increasing protein synthesis, we also want to make sure we're decreasing protein breakdown. So hopefully as a whole, you kind of appreciate now what it means to be an anabolic hormone. Insulin is increased when there's excessive carb uptake in the diet. And because carbohydrate represents such an easy source of energy, we're going to use that energy not only to store it as glycolysis, but also to store it as fat and to build protein. Now, there is one more thing I want to mention. This is something we still don't understand very well today, although there's been some good studies recently in the last 5 years to help us better understand the role that insulin plays in the brain. It's still debated whether the insulin affecting the brain comes from the pancreas, the peripheral insulin that we're going to be talking about, or whether it's synthesized directly in the brain. We still don't have a good idea of that. But it is becoming clearer that in the brain, insulin is regulating nutrients, and likely through this regulation of nutrients, is showing to play a key role in things like memory and cognition. As such, it's not too surprising to see correlations between obesity, type 2 diabetes, and an increased risk for things like dementia, even Alzheimer's. But again, what insulin is doing in the brain is still a bit of a mystery. There's ongoing research, but it's something that we're just not quite ready to wrap our heads around yet. All right, from a functional point of view, really the thing on this list that we care about the most is how insulin regulates glucose transport. And so let's take a minute to discuss this. As a hydrophilic protein, insulin is going to signal through extracellular receptors. Let's draw our cell membrane. Here's our insulin ligand. And as it comes upon skeletal muscle or adipose, it's going to bind to a class of receptors called FGF receptors. These are receptor tyrosine kinases. While we don't want to detail exactly how these types of receptors work, there are a couple of motifs that I'd like to share with you. For receptor tyrosine kinase signaling requires a process called dimerization, where two receptors come together. So we'll show our first receptor, and as insulin comes in and binds, this actually recruits a second receptor. So here's the receptor over here, but now it sees a docking site where that insulin is, and it's going to come move over and bind. That's dimerization. And once these two receptors come together, that leads to a signal cascade inside the cell. A cascade of events where one thing leads to another, leads to another. These are phosphorylation cascades. And while we haven't completely worked out all the minutiae of this signaling pathway, what we know is that this is going to lead to exocytosis. So we have exocytic vesicles, and these vesicles are preloaded with a carrier called GLUT4. And this transporter of all the GLUT members has a high glucose affinity. So when this vesicle is exocytosed, we'll see fusion of the two membranes, and that's going to lead to the expression of GLUT4 on the surface of the cell. And now that we're permeable to glucose, glucose follows its concentration gradient into the cell. Once inside, hexokinase grabs it, and using ATP, which will be converted to ADP, glucose is transformed into glucose 6-phosphate. Now it can't leave, it's trapped. There's a lot of things we can do with it. We can run it through glycolysis, we can make glycogen, we can turn it into certain amino acids, we can even turn it into the 5-carbon ribose sugars to make DNA, lots of different things we can do with it. Now we've got it inside the cell. And of course, this is what's lacking in someone with diabetes, either type 1 or to some extent type 2. In type 1 diabetes, insulin is not present, and so there's no way to move glucose into the cell, and that leads to very high blood glucose levels, and on the inside of the cell, a reliance on beta oxidation and protein metabolism for energy. And this is where the risks of ketoacidosis and things like that come from. Type 2 diabetes is a little bit different. There are aspects of this signal transduction cascade that are just sensitive to high levels of energy. And essentially, they just kind of shut down so that this process doesn't happen very efficiently. So even though we do have insulin, the cell has become insulin resistant, and we don't see the upregulation of GLUT4 on the surface of cells that would normally allow glucose to enter the cell. Now, speaking specifically for type 2 diabetes, what does that do? Well, in my opinion, one of the key flaws of diagnosing type 2 diabetes is looking at blood sugar. And in reality, blood sugar is not the problem. It's not a causal problem. It's merely a manifestation of something else. But with that heightened blood sugar, what we see are the manifestations of hyperinsulinemia. Now remember, insulin is an anabolic hormone, and if I have a lot of it all the time, that's going to have an effect on my body. That effect can be summed up in a specific disease state that we call metabolic syndrome. Now, in metabolic syndrome, there are 5 things that I'm looking for: increased blood glucose, abnormal obesity, increased triglyceride levels. That unfortunately leads to a decrease in high-density lipoprotein levels and an increase in blood pressure. The blood pressure is usually associated with increased sodium absorption in the proximal convoluted tubule in the kidney, increased water retention, things like that. Of course, the obesity comes from the increased triglyceride formation. It's this combination of things right here, symptoms, that we classify as metabolic syndrome and are associated with all the leading causes of mortality today. All right, there's one more thing that I'd like to talk about, and that is how beta cells in the pancreas are able to sense blood glucose levels and respond accordingly to those levels by releasing insulin. All right, so here's a beta cell. And of course, endocrine signaling is all about blood, so now we've got a blood supply. Of course, the trigger here is glucose itself. So as glucose levels are elevated in the blood, the transporter that I use on these beta cells is GLUT2. GLUT2 is different from GLUT4 in that it has a low affinity for glucose. And with this poor selectivity, that means it only works with high glucose levels. So as glucose levels increase, now that glucose is able to make it inside the cell. Of course, once inside the cell, what do we do with glucose? We run metabolism. And of course, the product of that is ATP. And this is where it gets really interesting. We have a potassium leak channel that's sensitive to ATP concentrations. Of course, potassium regulates resting membrane potential, and we need a constant flow of potassium out of the cell in order to maintain resting membrane potential. But because this channel in particular is sensitive to ATP, increased levels of ATP block or inhibit this channel from functioning. In other words, we stop the flow of potassium leaving the cell. So that has the overall effect of decreasing potassium permeability. And of course, if potassium permeability is decreased, that's going to lead to depolarization. And that depolarization is going to open up voltage-gated calcium channels. And of course, as calcium comes in, calcium does what calcium always does. This is exocytosis. I've got vesicles loaded with insulin, ready to be exocytosed. Calcium will fuse those membranes, and that insulin is released into the blood. Now, one thing we haven't talked about yet is glucagon and how glucagon, in many ways, is antagonistic to insulin. The signaling mechanisms are actually very similar, so we don't need to detail those. But let's see if we can sum this up in a cartoon. So insulin, it's a protein hormone. It's got a fairly short half-life of around 5 minutes. And remember, it's mitogenic, meaning it causes cells to grow. Now, the 3 key ways it's doing this are acting on skeletal muscle, adipose, and the liver. For both skeletal muscle and adipose, this is increased glucose uptake. And then we're going to either use that glucose for energy to do other things, or we're going to store it in the form of glycogen. And in the liver, increase glycogen production and decrease gluconeogenesis. I already have enough. I don't need to make more. And remember, the trigger for insulin release is increased blood glucose levels, or simply eating glucose. Carbs, if you remember from last unit, will release GIP. It's an incretin, and incretin will act on the pancreas to cause insulin release. The flip side of all of this is glucagon. Glucagon's primary effect is on the liver. However, recent studies have shown an effect on adipose as well, where we get increased lipolysis, and that's going to lead to increased free fatty acids in the blood, which, if I'm not eating sugar or don't have sugar in my blood, that becomes my source of energy primarily for my skeletal muscle. And in the liver, we're kind of antagonizing everything that insulin did. So in the liver, we would expect to see increased gluconeogenesis and the breakdown of glycogen in order to maintain blood glucose in a healthy state, blood glucose levels. And of course, the trigger for glucagon would be decreased blood glucose. And so from this figure, we can see that throughout the day, glucose homeostasis is regulated minute by minute, hour by hour, by a balance between insulin, if I've digested carbs, and glucagon, if I've not digested carbs. If insulin goes up, we're storing sugar and we're getting sugar into skeletal muscles so that we can maintain blood glucose levels. If blood glucose is down, then glucagon acts on the liver to increase blood glucose levels by increasing the breakdown of glycogen and increasing the production of glucose through gluconeogenesis. It's also working on adipose to increase lipolysis to provide skeletal muscle with energy in the absence of glucose.